Biology 1, this is your lesson on the cell cycle, which is going to include two types of cell division, and that's going to be meiosis and mitosis. And I'm going to talk to you first about interphase, which is where G1, the S phase, and G2 actually take place. And in G1, you're going to find that the cell grows in size as indicator that we need to start cell division. And once we get that indicator, we're going to go through synthesis, or the synthesis phase, which means that DNA replication is taking place. So it's in the S phase, that DNA replication, which means that DNA is copying itself during the S phase of interphase. And then the last part of interphase is going to be G2, which means that the cell's increasing in size, giving more of an indication that we are ready for cell division. As I mentioned, we have two types of cell division. That's going to be meiosis and that's going to be mitosis. And I am going to cover with you meiosis first. And I like to do that because meiosis has two parts. It has meiosis 1 and meiosis 2. And in meiosis 2, it looks exactly like mitosis. So if you learn meiosis 1 and 2, then you already know what the phases of mitosis look like. So let's start with meiosis. We've already talked about interphase, where we have the G1 and S phase and G2. We're always going to start the cell cycle with interphase. Interphase is always going to happen first, and then we're able to move into cell division. But the reason why we're able to move into cell division is because of gene activation. There's some genes that are turned on that tell us what kind of cells we need to produce. And in this case, we're going to be talking about the production of sex cells. Meiosis is the way that we get sex cells. This is the way that we get sperm and egg. And so we go through interphase, the genes are activated and telling us that we need sex cells, and we've already gone through interphase. And so now what we need to do is what we call prophase 1. And prophase 1 is the stage in meiosis in which the spindle fibers are going to form. Also, what's going to happen is the nuclear envelope is going to disappear, and the actual chromatin, or the chromosomes, are going to become more visible. So it's going to be during prophase 1 that the nuclear envelope is going to disappear. That means that the chromosomes are going to become more visible, and we're going to have the appearance and development of spindle fibers, which have a role in making sure our chromosomes go where they need to go. They're transport tubules that allow us to have our chromosomes go where they need to go in the cell. So what actually happens is during prophase 1 is something called crossing over. Crossing over is an event that happens between chromosomes in which they actually touch each other. And when they touch each other, they exchange genetic material. They actually change the genetic material in such a way that it causes there to be some variety in how we look, how we appear. It is through prophase 1 that you and I look different. It's through prophase 1 that you and I, though we may resemble our parents, our mother and father, we still look different from them. And we also look different from them genetically. So it is going to be through prophase 1 and crossing over that we get genetic variation, and therefore it's going to be through meiosis that we get genetic variation. So it's through meiosis that we get gene variation, or we get differences in genetic expression. The other thing that is going to happen once we are done with crossing over in prophase 1, we will move to metaphase 1. And in metaphase 1, those sister chromosomes, which means you're going to have four arms of genetic material that are attached to the chromosome are going to meet up in the middle of the cell. Meta means middle. So basically what those chromosomes are doing in metaphase 1 is they are meeting up in the center of the cell. And so once they do that, then we're able to move to anaphase 1. And in anaphase 1, this is where the sister chromosomes split or go in opposite directions. Ana means different. It means opposite. So basically what happens is those sister chromosomes are moving to opposite poles or directions in the cell. And so once they separate, what we're able to do is actually see that cell begin to divide, and that is going to be in telophase 1. And in telophase 1, the cell begins to divide, and you begin to see something that we call cleavage. And cleavage is when the cytoplasm begins to pinch or tear away from each other so that we can get two separate cells. And at the end of telophase 1, when that cell completely splits, it is called cytokinesis. Cytokinesis means to split or tear the cytoplasm. So basically, during cytokinesis, the complete cytoplasm has been split, and we have two different cells at the end of meiosis 1. And so then as a result of meiosis 1, we're able to move into meiosis 2. And in meiosis 2, we don't repeat interphase again. Interphase always only happens once. So we move into prophase 2. And so in prophase 2, it's very similar to prophase 1 in that the spindle fibers are going to form, the nuclear envelope is going to disappear, and the chromosomes are going to become visible again. But prophase 2, we're not going to have any crossing over. So crossing over only happens in prophase 1. Let me say that again. Crossing over only happens in prophase 1. So what we're going to get in prophase 2 is the disappearance of the nuclear envelope. We're going to get the formation of spindle fibers, and our chromosomes are going to become visible. And then we're able to move to metaphase 1. And in metaphase 1, the sister chromatids will meet up in the center of the cell or in the middle of the cell. Let me say that one more time. In metaphase 2 is going to be where the sister chromatids, that means half of the chromosomes, are going to be meeting up in the center of the cell. And so you should be able to see the difference between the two, because you should only see that there are sister chromatids that are meeting up at the center. And so don't get it confused, because in metaphase 1, we're going to have the chromosomes that are meeting up. And then in metaphase 2, we're going to be seeing that the sister chromatids are going to be meeting up. And once those sister chromatids meet up in the center of the cell, we're able to move on to telophase 2. And in anaphase 2, we're moving on to anaphase 2. And in anaphase 2, the sister chromatids split and go to the opposite poles of the cell or opposite sides of the cell. So in anaphase 2, the sister chromatids are going to move to opposite sides of the cell. And once that happens, we're able to go through telophase 2, which means that again, the cytoplasm begins to pinch away from one another, which we call cleavage, begins to tear away, and we begin to see two separate cells. And then in cytokinesis, we have two more completely different cells after meiosis 2. And so in the whole of meiosis, you are going to see 4 cells in the end. And all 4 cells look completely different because of genetic variation that was caused in prophase 1 through crossing over. So all of those cells are going to look completely different after meiosis. Meiosis only occurs in sex cells. It only occurs in sex cells. Let me say that one more time. Meiosis only occurs in sex cells. Then after that, because we're talking about sex cells, there are 2 types of cells that we can get through meiosis. That's going to be the sperm and the egg cells. And so whenever we get sex cells like sperm from meiosis, it's going to go through what we call spermatogenesis. Basically, we're getting the formation of 4 different sperm cells. And then in women or in females, we're going to get egg cells, and how we get that is through oogenesis, which means we're going to get 4 different eggs from meiosis. So remember, it's going to be through meiosis that we get our sex cells like sperm and egg. Now, when we go through fertilization, when our mommy and daddies had sex with each other, they formed, and we got you. It was your daddy's sperm and your mom's egg that came together, and when they did, they formed something called a zygote. And so you need to pay attention to this, because whenever you have meiosis, you always start off with a diploid cell, which means that there's going to be 23 sets of chromosomes and 46 chromosomes total. Let me say that one more time. Meiosis always starts off with a diploid, which means that we have 23 sets of chromosomes and 46 chromosomes total. But whenever we finish the meiosis process, we're going to end up with haploids. So that means that we end up with half of the sets of chromosomes and half of the number of chromosomes, which means that the haploids are going to have 23 chromosomes, which also means that your mom's egg had 23 chromosomes. Your dad's sperm had 23 chromosomes. And when they came together, they made a zygote, which is you, which now has 46 chromosomes. So I'm hoping that makes sense to you. So now with that in mind, as I mentioned before, if you commit to learning meiosis 1 and 2, and you pay very close attention to meiosis 2, you already know the phases of mitosis. So for the sake of time, I'm not going to go through all of the phases of mitosis. I am going to zero in on a couple other things that you need to be mindful of as you're looking and we're talking about chromosomes and cell division. Just like in our genes or in our DNA, we can have mutation. Remember, we talked about that. We talked about the 2 types of mutation that can happen during protein synthesis. Mutation can also happen in cell division. And one of the kinds that you need to be familiar with is something called non-disjunction. And non-disjunction is basically when chromosomes do not separate properly in anaphase. And in meiosis, it can be either in anaphase 1 or anaphase 2. But the thing is, non-disjunction is when chromosomes do not, or sister chromatids do not split crop properly during anaphase 1 or anaphase 2. This results in Down syndrome, and this ends our lesson on the cell cycle and cell division. Thank you.